दे रही हूँ। Excuse me, sir. Actually, I'm confronting a problem. Please don't mind.
Aksa, you should click to share your screen and navigate your presentation. Where have you downloaded to in your computer?
Yes, and that's downloaded in the computer. That's on the desktop. So how should I share it now?
On the screen below, there is a button. Name is shared screen. You should
Yes.
click once on this button. After that, it will ask where is your presentation, and you can navigate the presentation. Which one you have already downloaded to your computer.
So sorry for being late. So is there any else presenter? I think it
Yes,
will take.
it's coming. It's coming. You should change your presentation. It has begun to sharing your screen, but you should find your presentation. Yes.
I think it's coming.
No, it's desktop. We waiting for a PowerPoint presentation.
So the screen has been shared, but where is the presentation? Okay.
Yep. You are on the right way. Okay. Here it is.
So is it on the screen?
Yes.
Can you see it?
We can see it. Okay.
Okay, thank God. Okay, today I will share the following contents. For example, first of all, introduction, then the research objectives, and then the material methods, and so on. So let me start it. In this research project, I used the alpha tocopherol, that's the vitamin A. It has various benefits for the physiological benefits on the fish health. For example, it can improve the spawning success, and it also prevents the peroxidation. You know, that's the oxidation in the body. That's the dangerous for the aquaculture animals. So vitamin A that prevents the peroxidation. So, and the third benefit of the alpha tocopherol, it can scavenge the free radicals. So it is also a determinant factor for the reproductive fitness because it determines the gonadosmetic index, the egg size, and so on. And the fifth benefit of the alpha tocopherol, it can enhance the gonadosmetic index. And in this research project, I used the corn gluten meal. That's a plant meal. It has been used in place of other plants or animal proteins because the fish meal, that's very costly. And in Pakistan, especially in the Government College University, Faisalabad, we are using the plant meals because that they are very inexpensive, and the fishes that also accept this kind of diet. And the second benefit of the corn gluten meal, its protein content ranges between 60 to 7% of the dry matter, and actually this is equivalent to that of the fish meal. So we don't have any protein deficiency problem in the fishes, and it has been used in the feed of various fish species. Then I use the labia rohita, also known as the rohu in the Pakistan. It is one of the major carps, and it is very economical fish because it has very fast growth and growth rate. It has good taste, which is the basic requisite for the culturing of this fish species. And my research objectives, I have three objectives in my mind. For example, to improve the antioxidant status of the labiorohita fingerlings, and the second objective was to analyze the nutrient digestibility of the labiorohita, and by feeding them the alpha tocopherol supplemented diet. And the third, that's I think the most important objective of my research, because the fish farmers, they confront with the budget problem. So our research is to provide the cost effective feed to the farmers. So can you hear me? Are
Now
you
we're here.
understanding?
Now
Okay,
we
okay,
are
okay. Thank
listening.
you. Okay, thank you. And these are the material methods which I performed, which I followed during the trial period. Actually, I conducted the 70 days feeding trial in the laboratory of the Government College University, Faisalabad, Pakistan. And first, I procured the fingerlings from the hatchery, Government Fish Seed Hatchery, Faisalabad. Then I stocked 15 fingerlings in each tank, and I gave them about 2 week period for the acclimatization to the laboratory condition, because you know that when we transfer the fingerlings um, uh, uh, from the from one environment to the other, then we should give them a time for their acclimatization. And during that period, I fed them once a day. I fed them once a day. So because the fish from the hatcheries, they may contain some fungal parasites, that's why I gave them the saline bath of 5 gram sodium chloride, 5 5 liter sodium chloride solution. And the experimental layer seven, I prepared 7 diets that were isoprotein, isocaloric, and isolipidic. It means they have all these contents similar, just I supplemented these diets with the alpha tocopherol at the graded levels, and these levels were the 100, 200, 300, 400, 500, and 600 milligram per kilogram. These diets, and then I fed these diets to the fingerlings for the period of 70 days. And during that period, I in the morning I offered them the diet, and preceding the 2 hour of the diet, I washed the tanks. It means that all such feeding trial was conducted in the university laboratory. Here it is the ingredients composition of the test diets. All the diets, these ingredients was added. For example, first of all, alpha tocopherol, and graded levels I've already told you. Then the corn gluten meal was also added. Same amount of the meal was added in all the diets. In short, I can say that all the diets were similar, having having 100 total its weight 100 gram. So 100% diet was prepared. Only the alpha tocopherol was different in them. Then I prepared the feed in the form of the pellets, because fish only accept the feed in the form of short pellets, short sized, and a size of about zero point 0.5 millimeter sieve size of the pellets was maintained during the whole diet. And then the homogeneous mixture was also prepared in the electric mixer for 10 minutes by adding fish oil. Fish oil was added gradually to maintain the consistency and to prepare the suitable dough. And to hydration at the rate of 10 to 15% was also done, and at the last, floating pellets were prepared through lab extruder. Feeding protocol for sampling that I've explained it earlier. I think I should move on. These three parameters were maintained during the feeding trial. For example, water temperature of about 20 to 30 degrees Celsius was maintained. Then the pH was maintained during between 6.7 to 7.7, and the DO, it means dissolved oxygen, was also maintained within the range of 8.68 milligram per liter to 10.92 milligram. And the pictures around the on the sides of the slide, first picture is showing the feeding plate on which the feed was added, and the second picture shows the tanks in the row in the laboratory, and the third picture is showing the mesh that was present in the tank, and the fourth picture is showing that are the valves which were opened and closed for the collection of fishes. Then growth study. I studied the growth of the fish by using the standard formula of National Research Council. Weight gain percentage was determined. FCR, it means feed conversion ratio, was determined. And at the third, standard growth rate percent was evaluated. Here, then I conducted the chemical analysis of feed and feces. The crude protein in the feed and feces were determined by microgeldal apparatus, and gross energy that was determined by oxygen bomb calorimeter. Crude fat was estimated by petroleum ether extraction method in the Soxtec system, and ash was determined by electric furnace. Here is the formula of the apparent nutrient digestibility coefficient, which was determined to evaluate the crude protein, crude fat, and gross energy of the fish. And the antioxidant status was also determined by determining the percentage inhibition of oxidation, because oxidation, that's dangerous for the fish species. So we want to antioxidize it by alpha tocopherol, and its results were determined in terms of percent inhibition. Of the percent inhibition is equal to absorbance of the control minus absorbance of the sample, and hence oxidation percentage was also evaluated. Statistical analysis, that was all the data was analyzed by ANOVA, analysis of variance, one-way analysis of variance at the COSTAT computer package, and then the means were compared by Turkey's honestly significant difference test at probability less than 0.05 value. So here is the results. So are you waiting for the results, sir?
Yes.
Okay. Okay. So here are the first results. These are the about the growth parameters, because growth is the most important parameter of the fish study. If any diet is not in the benefit of the growth, then it is not recommended for the future use. So growth parameters, first of all, that's the initial weight. Initial weight of all the fingerlings was more or less similar. That was about 9.08 gram of the fish. Then the second row shows the final weight of the fish. The final weight is actually I will tell you about the most striking results of this research. It was deduced that 200 milligram of the alpha tocopherol that brought about the most striking results in terms of growth. The weight gain and the weight gain percentage, that is marked by the blue circle. By adding 200 mg milligram of the alpha tocopherol brought about the results Weight gain of the fish was 23.67 gram. Initial weight was 9 gram, and the final weight was 23 gram. And weight gain percentage was 261%. I think that's the big achievement of my research. And after the test diet 3, the good results were obtained by adding the test diet 4, having 300 milligram of the alpha tocopherol, and these results were the 20 gram 1 gram of the weight gain and 232% of the weight gain. And if we discuss about the FCR, that is the feed conversion ratio, then the feed conversion ratio, the best results were obtained by adding the test diet for having 300 milligram of the alpha tocopherol, while the negligible improvement in the weight gain was observed by adding the test diet 6, having 500 milligram of the alpha tocopherol. And so the next, we'll move on the analyzed nutrient composition, because first of all, feed nutrient composition was determined, then feces nutrient composition, and at the last of the fish. In the table 2, nutrients composition is present, and all the nutrients composition was more or less similar in all the diets. It means there is somewhat negligible difference in the nutrients composition of the feed. And here it is the composition of nutrients in the feces which were collected from the tanks after adding feed. So these are the feces nutrient composition, and most important is the apparent digestibility coefficient, which is the parameter to find the crude protein, crude fat, and gross energy in the fish meat. So here the test diet 3, having alpha tocopherol level 200, brought about the most striking results, good results in the form of crude protein. Crude protein in the fish was 73%, as compared to, it is significantly different from other treatments, and as well as with the control. And crude fat was 74.48%, and the gross energy was 63.61, and it is best results than the other treatments at the probability level 0.05. And then these results were followed by the next higher treatment, the 300 alpha tocopherol. Actually, the trend here is that the increasing trend was observed till the level 200 mg level of alpha tocopherol, and then decreased level of improvement was observed in other treatments. And at the last, the antioxidant activity. Here the treatment number, experimental diet number 7, having alpha tocopherol level 600 mg per kg, brought about the good results in terms of percentage of oxidation. Only 7% oxidation occurred in the fish meat, while you can say this value increases as the alpha tocopherol, it means vitamin E level decreases. So we should add 600 mg per kg of the alpha tocopherol in the diet to prevent the oxidation. And now here it is the conclusion. So the first conclusion of my research was that it means that supplementation of alpha tocopherol, vitamin E, it played a major role in improving apparent digestibility coefficient of nutrients, antioxidant activity, and growth performance of Labiorhita fingerlings as compared to the control diet. The second conclusion was that 200 mg per kg, that's the optimum level, that's the level that we define that the farmers or the future studies should incorporate this level for the maximum results in the fish performance. And furthermore, supplementation of alpha tocopherol in corn gluten meal based diet provided us economical and eco-friendly feed for fish. So that's all from my side. These are the references I used. So any questions, sir?
Okay. Aksa, thanks for that nice presentation.
Thank
Dr.
you so
Zeki,
much.
if any participation wants to make a comment or ask any question, they can, there is a button below
Yeah,
the
I know.
screen, reaction button, and
So
click to thumbs up, please.
thumbs up, or I should make them unmute? Hold on.
If there are any question, just click to thumbs up and we can make them unmute.
So, okay. If there is a, are there anyone with questions to Aksa? I think there is no questions.
No question means that all of the lecture was understood or nothing was understood.
I should remind something, Aksa.
Yes, sir.
In results section in your table 1,
Yes.
go to table 1 and see you use canola meal instead of corn meal.
Oh,
You just need to change the title of table as corn meal instead of canola meal. Okay?
yes. Yes, sir. I noted.
Okay, if there is no questions, I should go ahead and pass to next presentation, and it will also be presented by Aksa, right? It is actually it's almost the same, but only the meals and the fish species are different, right?
No, sir. Supplementation is also different.
Different?
Yes, that's the
Okay.
nanoselenium particles.
Okay, our other presenter is also here, Aksa. If you wish to take some rest, I will go ahead and give the speech to Sadia Zafer, or you can go ahead and present the third paper also.
I think I should
Go ahead and
take.
present. Okay.
Yes, yes.
Okay. The third paper is role of nanoselenium particles supplemented sunflower meal-based diet on growth performance, nutrient digestibility, and hematology of Chirinus marilla fingerlings break. Okay.
Sir Highness Meregala.
Go ahead and share the next presentation with us, Aksa.
Yes, sir. So can you watch it now?
No, we can see now.
You should do the same things as you did for the previous presentation.
Okay.
You should use share screen option and share your presentation with us.
Yes, it's
Okay, it's coming now.
Aksa, could you please make it full screen, please? Okay.
Yeah.
Okay.
Yeah, top of the screen. Yes, that's it. Thank you very much.
Is it obvious now?
It is okay now.
Okay.
We are listening.
So I'm again the Aksa Sharif.
We already know you.
Okay, okay. I think I should skip my introduction.
Yeah, I think.
Okay. And the second presentation is about the supplementation of the selenium nanoparticles and its effect on the growth, nutrient digestibility, and hematology of Cirrhinus mirigilla fingerlings fed on sunflower meal-based diet. So to take a start, I'll tell you about the fish farming. As we know that fish farming, that's the best industry to incorporate and commercialize the nanotech products. The products that are the nano sizes in the form of about less than 100 nanometer. So but unfortunately there is very scarce work in this regard. So nanotechnology tools that are being used for rapid disease reduction and targeted delivery of nutrients in aquaculture, and they also increase the bioavailability of nutrients and minerals to the fish. The particle size of minerals in nanoparticle is smaller than 100 nanometer. If someone asks that what's the difference between a normal size particle and the nanoparticle, then the answer is that nanoparticle that are of the size of less than 100 nanometer, and we use these particles because they can easily pass into the body cells as compared to the ordinary minerals. And we use the selenium, that's the trace element in the in the fish body, and that's responsible for various physiological functions. For example, it is involved in the enzyme activity and prevention of oxidative damage to body tissue, as the alpha tocopherol also prevents from the oxidation. And in the same way, selenium nanoparticles also prevents oxidative damage to body tissue, and its deficiency create great loss in livestock. So the second, there are also some other functions of the selenium. That's why I selected this trace element for my study, because it maintains the thyroid hormone metabolism, cell growth, and antioxidant defense system. So it is also required for the growth, fertility, and immune system of animals. And there are several references that its supplementation in the basal diet can improve the final weight and relative gain rate of crucian carb. So then why I use the sunflower meal? Because it is the most appropriate protein source, because its protein content is in the range of 34%, and it is also easily available as compared to the fish meal or any other meals. Actually, for the fish farmer, that meal is the most appropriate, that is easily available, that's economical, and that is in the good supply, that shouldn't be in short supply. So I use the sunflower meal. And the third, that's why I use the Cirrhinus mirigilla. That's one of the Indian major carp. It is also known as the gulfam and mori. You can also that's the common name of this fish. It have high growth rate and good taste. And when we use this fish in the polyculture, then its feeding habit is the bottom feeding fish that feed on the detritus present in the bottom of the water body. Then what were the objectives? My research objectives were to estimate the effect of selenium nanoparticles on growth performance of Cirrhinus mirigilla, and then to determine the effect of these particles on the nutrient digestibility of the Cirrhinus mirigilla, and the third, to determine the hematological parameters, that the hematology that's related to blood parameters, and at the last but not least, to determine the optimum level of selenium nanoparticle supplementation in plant meal based diets. And in the same way, these are the material and methods which I followed for this uh, study. Then first of all, sir, should I repeat the material methods in this now?
I think you can pass this section. Because
Sorry.
you use the same materials and same experimental setup, right? With the
Yes, yes,
previous study. So
yes, yes.
you can pass these parts fast.
Okay, okay, okay. But I think I must highlight this slide because that's the experimental layout that
Uh-huh.
the selenium nanoparticles were used in these concentrations 0.5 mg per kg, then the 1 milligram, then 1.5 milligram, 2 milligram, 2.5 milligram, and 3 milligram. Total 7 diets were prepared, and 0 milligram was added in the control diet.
Mhm.
And here the
Baby
experiment.
images.
Yes, you are you already know this
Yeah,
experimental
real.
setup. Okay. Then the feed ingredients were collected, and then the feed pellets were prepared, having the sieve size 0.5 millimeter, and all the 7 diets were supplemented with the graded levels of nanoselenium. Water was also added in the feed mixture to prepare the suitable dough. Then here are the ingredients composition of the test diets, and that's obvious that nanoparticles. I already told you the levels of the nanoparticles, and then the sunflower meal was added 50% of the in each test diet, and then other ingredients are also similar. And here the nanoparticle stock solution was prepared. The preparation and confirmation of stock solution was carried out according to the Frederici and Ramsden by following their methods. And briefly, stock solution of at least 99% nanoparticles. About maximum impurity should be about 1%, and an average particle size of less than 100 nanometer was made. From the stock solution, further dilutions were made to ensure above said dilutions. And additionally, stock solution of 90 nanoparticles was made by dispersing the nanoparticles in ultra pure water with sonication for 6 to 8 hours. Here is the feeding protocol. Feed was added at the rate of 5% of live wet weight. Then the 15 fishes were stocked in each tank, and the feces were collected by opening and closing the valve 1 and valve 2 of the tanks. And the feces that were collected from the valves, they were dried in the oven and stored for chemical analysis. Chemical analysis of feed, feces, and whole body analysis. Whole body analysis, and the same methods were again followed. Moisture was determined by oven drying at 105 degrees Celsius for 12 hours. Then the crude protein was determined by microgel dilatators. Crude fat was determined by petroleum ether extraction method. Crude fiber was determined as loss on ignition of dried lipid-free residues after digestion with 1.25% sulfuric acid and 1.25% sodium hydroxide. Similarly, ash was determined and gross energy by oxygen bomb calorimeter. And in the same way, I calculated the apparent digestibility coefficient by using the formula that's obvious on the screen. Then I determined the growth of the fingerlings of Sirhinus miracula and their weight gain percentage, their feed conversion ratio, and their standard growth rate was determined by manipulating the data in the following formulas. And the hematological study. To study the hematology of the fish was anesthetized with clove oil, and then the heparinized syringe was injected into the caudal vein of anesthetized fish to collect the blood, and that blood was taken to the molecular lab, Department of Biochemistry, University of Agriculture, Faisalabad, and hematocrit that was determined by microhematocrit technique. Hemocytometer was used to determine the number of cells of the RBCs, red blood cells, and the white blood cells, and hemoglobin that was determined by the method of the Wedemeyer and Yestuge. And statistical analysis, the data of the growth, nutrient digestibility, uh, mineral absorption, carcass composition, and hematological indices of fish was subjected to one-way analysis of variance ANOVA. And the differences among means were compared by Tukey's honesty significant difference test and considered significant at probability less than 0.05. And the CoStat computer package was used for statistical analysis, having version 6.303. And to illustrate the results, here is a figure 1. It illustrates the size and shape of selenium nanospheres by transmission electron microscope. That's the view of the transmission electron microscope. And in the figure 1, the 10 nanometer scale bar was set. Actually, this photograph show you the size, confirmed size and shape of the particles which I used in my study. And in the figure B, 50 nanometer scale bar was set. Here is the figure 2. And actually, that's the X-ray diffraction photograph, pictorial view of the crystal structure and phase composition of selenium nanospheres. It confirms the crystalline structure of the nanoparticles, and we compared them with that of the standard size of the nanoparticles. And there was a strong, you can say that similarity between these two structures, crystalline structures. And the growth performance of Cirrhinus mirigla fingerlings, growth performance, to elaborate this table, I will say that the weight gain percentage, and the standard growth rate that were good, that were obvious by adding the test diet 3 having selenium nanoselenium particles at the level of 1 milligram per kilogram. We can say that if we add 1 gram of selenium nanoparticles in the diet, then we will get the good weight gain ratio, which is 12.31. The initial weight of the fish was 10.24, and the final weight of the fish was 12.31 by adding 1 milligram of the selenium nanoparticles, and the weight gain percentage was 189%. And with the control diet, you can maintain take the difference that by adding the control diet, the weight gain percentage was 157%, while by adding 1 gram of selenium nanoparticles, then this weight gain improved to the 189%. So you can you note this difference? Hello, sir. Can you hear me?
Yes, we can all hear you.
Okay. And the feed conversion ratio, that's also highlighted in the red color, feed conversion ratio, that's also 1.58. It means that it tell us how much of the feed is converted into the fish body. That when the feed is given to the fish, then some part of it the fish intake, and while the other that's get wasted. So the FCR tell us that how much of the fish feed that has been utilized by the body. So 1.58 gram of the fish feed was utilized, and the survival rate of the fish was 100%. It means no any fish was dead in the tank, having test diet 3. And the second improved results were second improved results were got by adding the test diet 7, having the mm, sorry sir, actually I want to say that the minimum level of the weight gain, that one is the control level and the other are the graded levels of the nanoselenium particle. First good results were obtained by adding the test diet 3, while the minimum, you can say that less good results were obtained by adding test diet 7, having 3 milligram of the selenium nanoparticles. And in it, weight gain percentage was only 156.79, and that's actually equivalent to that of the control. It means there isn't any significant difference between the control diet and the test diet 7, having 3 mg per kg of selenium nanoparticles. Then the percentage of nutrients in test diets was also determined, and more or less similar nutrients were present in all the diets. Crude protein was about 31.4%, crude fat was about 6.94%, and gross energy was about 3.87% in all the diets, more or less similar. And that's the percentage of nutrients in feces of Cirrhinus mirabila fingerlings. In feces, the crude protein was crude protein was present in the feces of the tank that were offered the test diet 3, and the crude protein was 9.36%. Crude fat was 1.9, and it means that most of the feed was utilized by the fish body, and a portion of it, a minimum portion of the feed was excreted out from the body. Here it is the percentage of nutrients digestibility of Cirrhinus mirabila. You can say that apparent digestibility coefficient. Again, the same results. The test diet 3, that the the good results were obtained by the test diet 3, having 1 milligram of the selenium nanoparticles, and crude protein was obtained was about 72%. That's the highest among other values, and also significantly different. Crude fat, that's also known as ether extract, crude fat was 73% in the fish meat, and gross energy was 67%. And these data values that are highest than the others as compared to control. You can make a difference between the control diet and the tested 3. In the control it was 54% crude protein, while in the tested 3 it was 72%. In the control, ether extract was about 51, while in that of tested 3 it was about 73%. And in the same way, gross energy was 49% by in the tank that was offered the control diet, while the 67% in the tank in the fishes that were offered with test diet 3. And hematological parameters of Cirrhinus mirabila fingerlings were also determined. First of all, RBCs, red blood cells, were highest in the blood of the fish treated with the tested 3, having 1 milligram of the selenium nanoparticles. White blood cells were also greatest in this fish, 7 cubic millimeter, and the platelets were 66. Hemoglobin was 8.48 gram, and the see, just a minute.
Aksar, try to use your time efficiently.
Yes, mean cell hemoglobin concentration, that was also highest in the 30 in the test in the fish treating with the test red 3. And the mean cell hematocrit was also highest that was the 50%, and mean cell volume, that was also highest in the by adding the test diet 3, that was 190%. The second good results were obtained by adding the test diet 4 having 1.5 selenium nanoparticles, while the minimum level of all these parameters were present in the tank treated with the test diet 7. And at the last, here is the conclusion of my study. Supplementation of the selenium nanoparticles that is helpful for the proper growth performance, nutrient digestibility, and improvement of hematological parameters of Sirhinus merigla fingerlings fed sunflower meal based diets. And it was also deduced that 1 mg per kg supplementation levels of selenium nanoparticles is the optimum level for the improvement of all above said factors, and higher supplementation did not cause further improvement. And here are the references.
Okay.
Thank you.
Again, we thank you, Aksa, for that nice presentation. And are there any questions from the other participants or contributions? I will go ahead and pass to the first paper to be presented by Sadia Zafar. No questions? Okay, then we are going to listen a presentation by Sadia Zafar on diversity and ecology of ants from district Naushara Feroze, Sindh, Pakistan. Okay, I think Dr. Sadia Zafar is here, right?
Yes, hi, I'm here.
Okay, first introduce yourself briefly, then start your presentation, Dr. Zafar.
Yes, hi everyone. I am Dr. Sadia Zafar and working as assistant professor in the University of Education, Lahore, Faisalabad campus in the Department of Botany.
Sadia, also start your video.
Can you see my screen?
We see your presentation, but we can't see your face. Start your video also.
I am trying, but I don't have access to share my video.
You don't have a camera?
I have camera,
Okay.
but they said you can't start video because you don't have access.
Okay. Just go ahead and first, as I said, introduce yourself briefly, then start your presentation.
Yes, sure. Bismillahirrahmanirrahim. I am Dr. Sadia Zafar, Assistant Professor in the Department of Botany, University of Education, Lahore, Faisalabad campus. And today I will talk about the effects of aspergillus flavus inoculation on grain shield and nutrition of maize, deer maize in spring and autumn season.
Dr. Zafar, could you please make this screen full screen? Thank you.
Now fine?
I think
Yes,
I gave you,
so
I think I announced the title of your presentation wrong, right?
Yes, yes.
Okay.
My topic of my presentation is the effects of aspergillus flavus inoculation on grain yield and nutrition of maize, deer maize in the spring and autumn season.
Okay.
These are the contents of my presentation. As we know that maize, that is also known as corn, which is also known as king of cereal, because it is very important and has very important value for nutrition. And the maize is also considered as third important cereal crop, and it is world's most widely cultivated crop. According to an estimate, maize is cultivated around 150 million hectares around the world with production 1012 MMT, and in Pakistan it covers 0.9 million hectare area with annual production of 1.3 million tons. Global maize production is under several abiotic and biotic threats. As we know that the crops that are facing many stresses and can compete all the stresses, and maize production is lost due to several biotic and abiotic stresses, and around 2 to 50% of the maize production is lost annually around the globe, and in Asia it lost about 12%. There are many stresses, and the biotic stresses are, and the effect of the biotic stresses are very severe, and the fungi are among the biotic stresses that affect many crops and loss the yield. And the fungi affect the maize and other very important crops, and many uh, genera and species of the fungi cause heavy loss of the nutrition as well as the yield of many important crops. The Aspergillus genus, which is a dominant genus of the fungi group that cause heavy loss around the globe, and the two important species of the Aspergillus, that are the Aspergillus flavus and Aspergillus parasiticus, that cause infection in several crops, and their contamination produce aflatoxins. As we know that aflatoxins are the compounds that are carcinogenic and are harmful for the several animals as well as for humans. Even they cause death in rare cases. And the optimum temperature for the growth of Aspergillus flavus is 37 degrees Celsius, and they cause aflatoxin contamination under high temperature and under high humidity. And limit of aflatoxin for human consumption is 4 microgram per kilogram for aflatoxin B2 type, and aflatoxin G1 and aflatoxin G2, while the 2 microgram per kilogram for aflatoxin B1. That means that aflatoxin B1 is more dangerous as compared to other types of the aflatoxins. And about 55 million people are exposed to aflatoxins at unhealthy level, and they cause several types of diseases and even cause death. The objective of my presentation was to assess the genetic diversity of Pakistani maize genotypes for recommendation of suitable maize genotype in terms of better yield and grain quality. Now I will discuss the material methods. The whole study was carried out in two growing seasons, in spring season and autumn season. And the experiment was conducted in the Ayub Agriculture Research Institute, Faisalabad, Pakistan. There are different types of the treatments was given during this study. A genotype into treatment pictorial arrangement was used by involving 14 different types of the genotypes that are available in the Pakistan and commonly cultivated by the farmers, and they are given with two treatments. And the fungus, which was the Aspergillus flavus, was inoculated, and the plot was divided into two equal parts. One that is treated with the aspergillus flavus, or in other words, aspergillus flavus was inoculated in the plants, in the cobs of maize that are named as inoculated group, and the another group that was not treated with the fungi and was named as non-inoculated group. There are maize genotypes which was used in the study. These different 14 genotypes, some are the hybrids and some are the varieties. Varieties like Pearl, Malka, and MMRI, these are the varieties, and other 11 are different types of the hybrids. We also take the meteorological data, which is important because aspergillus growth grow under optimum or favorable conditions, like they favor the high temperature and high humidity. After inoculation or giving the treatment, the grains was calculated and the cobs were harvested from the plants of maize, and cobs weight, cobs length, number of grains per cob, 100 grains per cob was measured. Mycological studies was also done for the confirmation of presence of the Aspergillus flavus species in the grains. Chemical analysis of the grains was also done, that includes the moisture contents, oil contents, total soluble proteins, starch, total carbohydrates, ash, fibers, vitamins, and amino acids. The data related to the yield related parameters and chemical analysis was statistically analyzed by the analysis of variance using Minitab 17 software. The significance was determined at 0.05 probability level for each treatment. And these are the results. Here are the presentation of some meteorological data during 2 different seasons of the of experimentation. One is spring and another is the autumn. Here we can see that during the first experiment, here the higher temperature as compared to second experiment and relative humidity was higher in second experiment as compared to first, and the rainfall was higher during first experiment as compared to second. Now I will discuss the results of first experiment. Here are the maize cobs, and the black circle showed the infection of the aspergillus flavus that are present among the grains, and that produce high amount of aflatoxins at the time of the storage. And these are the cob length of different aspergillus species, and there was non-significant difference among the different uh, uh, varieties of genotypes of the maize, but there are the significant difference among the treatment. And the cob diameter was not affected by the inoculation, as we can see in the picture that there are not as difference in the different genotypes, and inoculated and non-inoculated has variable results towards the inoculation of aspergillus flavus. And the cob weight was affected by the inoculation of aspergillus flavus. It means aspergillus flavus inoculation has effect on the cob weight. And the number of grains per cob was highly affected in almost all of the genotypes except 2 genotype of the maize. That means that aspergillus flavus affect the number of grains of per cob that reduce the number of grains per cob. And total grain weight per cob also reduced by the inoculation of aspergillus flavors. 100 grains weight also decreased by the infection of aspergillus flavors. Oil contents also affect. It means the quality or nutrition of the grain also affected by the infection produced by the aspergillus flavors. The proteins and the moisture contents also affect by the inoculation of aspergillus flavors. But the starch has variable response towards the aspergillus flavors inoculation. And the fiber contents also decrease in inoculated group as compared to non-inoculated group. Ash contents are also decreased in almost all species, all genotypes of the maize after treatment with aspergillus flavors. And the carbohydrates also decrease in inoculated group. And total free amino acids also reduced by the contamination produced by aspergillus flavors. And the results of second experiment which was carried out during autumn season. Here we can see that there are many cobs which show the infection produced by the aspergillus flavors. During second experiment, during autumn season, cob length was affected by the treatment, but most of the genotypes have variable response toward the treatment. Or in other words, we can say that cob length don't have effect toward the aspergillus flavors inoculation, and the cob diameter also had no effect or variable response toward the treatment. And cob weight in many genotypes reduced after receiving the treatment of aspergillus flavors. And inoculation of the fungi also decreased the number of grains per cob in second experiment also, as it reduced in first experiment. And total grain weight per cob also reduced in all species during a second experiment. 100 grain weight also reduced by the treatment of aspergillus flavors. Here are a pictorial representation of aspergillus flavors that was confirmed from the grain and was identified by morphological study and by microscopic studies. And oil contents all had variable response toward the inoculation of aspergillus flavors, but in most of the genotypes of maize, they reduce the oil contents. Moisture contents also had variable response towards the treatment, as it increased in some inoculated, while in other genotypes, the moisture contents increase in inoculated or non-inoculated group. And the carbohydrate was reduced uh, for the treatment of aspergillus flavors. But the starch has variable response and had different response for each genotype. Some genotypes have
Could you
increased
please
and
get
some
faster?
genotypes have decreased. Yes, sir?
Could you please get faster?
Yes. And the fiber contents also had variable response, and ash content are also variable for aspergillus flavors. The protein contents in the second experiment was higher in inoculated group as compared to non-inoculated group. Total free amino acids were also higher in non-inoculated
Dr.
group.
Zafer, excuse me, Dr. Zafer, decision going to power off automatically after 2 seconds.
2 minutes.
Yes,
Yes,
and
yes.
conclusion, non-wounding aspergillus flavus inoculum affected the yield and quality of maize genotype under high temperature. During spring season, maize genotype produced much higher aflatoxin as compared to autumn season. Under high temperature, the aflatoxin production was relatively higher in first experiment as compared to second experiment. And there are some recommendations. KSC 9663 performs better in terms for shield during spring season, and we can choose the FH 1046 for autumn season that shows more resistance and can give better shield. Thank you so much.
Okay. We thanks Dr. Zafer for your nice presentation. And I think I have to finish up this session quickly because the next session is almost about to start, right?
Thank you so much.
Okay. I thank you all participants and the presenters. And I wish a successful and beneficial conference to organizing committee. Okay, thanks for your participations.
Thank you for your attention, Dr. Gökalp. Thank you so much.